Hi everyone, my name is Matthew LaFauve, and I am a postdoctoral fellow in the Institute of Environmental Health Sciences at Wayne State University. Today, I'm thrilled to tell you all about a portion of my dissertation research looking at various aspects of neuroanatomy in invasive Asian carp. So I'm going to start with some acknowledgments to my colleagues in the Hernandez lab and to my colleagues at the United States Geological Survey for helping me with this work and for, most importantly, keeping me excited about the brain in all of these fishes. I also want to say thank you to the Animal Behavior Society for putting on yet another incredible virtual conference. So just so that we're all on the same page, let's talk about the fish brain for a moment. Starting at the anterior or front portion and moving posteriorly or backwards, the telencephalon includes the olfactory bulbs, and the main body structure houses the pallium, primary olfactory processing, and most social cognition. The diencephalon is the primary neuroendocrine region of the brain, housing the hypothalamus and the main relay station, similar to the thalamus in mammals. The mesencephalon is integral for multiple sensory integration and processing, and is also responsible for alertness. The hindbrain is divided into the metencephalon, which houses the cerebellum, helping with movement coordination and learned motor behaviors, and the myelencephalon, which houses the vagal lobe in some fishes, which is responsible for taste, and is the primary relay station for information coming from the spinal cord to the brain. So we've just gone through the brain, pointing out some structural regions that are known to have functional specializations, most importantly for this concept of neuroecomorphology. Their structural regions may have huge implications for ecological niche habitation and for organism function in the diverse aquatic environments on our planet. If we take a general fish brain, and for our purpose today, we can think of the zebrafish brain as a general fish brain, different environmental pressures, such as deeper water or ocean open living, are going to change how the brain looks and functions. This is related to environmental pressures for detecting and releasing signals that best propagate in those environments. In addition, fish tend to exhibit a use it or lose it pattern. This means that if they live in an environment where they don't need such a high degree of sensory detection and processing, that part of their brain may reduce in size to avoid the energetic cost of nervous tissue. This is exemplified by cave fish. They've secondarily lost their eyes, and the region of the brain that processes vision, indicated here as TEO, is much reduced in size. So as the environment changes, brain structures may as well. This brought me to an overarching question of my research, which is, what can the brain tell us about how an organism may function or functions in a new environment? Working with the United States Geological Survey has allowed me to begin to ask this question in an important system, the invasive Asian carp. These three species are making their way up the freshwater river systems in North America and threatening fisheries in the Great Lakes and beyond. Silver carp and bighead carp, together called the bigheaded carps, are much more closely related to each other than they are to the grass carp. In addition, the bigheaded carps are filter feeders, sifting out phytoplankton from the water column for food. Grass carp, as their name suggests, eat vegetation growing in and floating on the water. All three carp species have slight differences in their sensory needs for success in capturing food, and may exhibit behavioral differences in both their native ecosystems and here in their invaded ecosystems in North America. In order to help piece together how any differences in brain morphology may result in species differences, I needed to know what their brains looked like. So I set out to establish an atlas of their brains to serve as a base understanding for future work in developmental neuroscience and neurophysiology in these species. Using specimens collected by the USGS, we used micro-CT scanning to determine gross brain morphology. This was done using the contrast agent phosphotungstenic acid, or PTA for short, allowing the CT scan to look more like an MRI, lighting up the soft tissues by differences in density. From these scans, I was able to isolate specific regions for further analysis and to generate a three-dimensional rendering of what the brain looks like from inside the head. Because this is a non-invasive method, there was no damage from removing the brain, something that is only recently possible in comparative neuroanatomy. This is what one of the micro-CT scans looks like, traveling from the front of the head towards the back. I could honestly look at this scan all day. It's oddly relaxing. The brain is a structure in the middle, right here. The benefit of the stained micro-CT scan is that it also highlights all the other soft tissues and hard tissues in the specimen. So coming back to what the software is able to generate after I segment out the regions, it creates something that looks like this. This multicolored structure is based on those segmented regions that I've just pulled out. So adding the head around the brain allows us to see a combination of the soft and the hard tissues to see really where exactly the brain sits in the head and how it's connected to the rest of the structures in the craniofacial region of these fishes. The colors for each of the regions are going to be consistent across the results. So when comparing the three-dimensional renderings between the species, we see that the big-headed carps, as a reminder, that silver and big head, are relatively similar when compared to the grass carp. There are slight differences in where the brain sits in their skulls, but overall, their gross morphology is relatively similar to each other. Overall, though, grass carp have larger telencephalons and mesencephalons when compared to the big-headed carps, while the big-headed carps have larger cerebellums and vagal lobes than the grass carp. So in addition to gross morphology, just based on looking at this, the three-dimensional rendering can also tell us about the volume of these brain regions of interest. The volume of the brains of the carp are a way for us to have a more quantitative aspect of that morphological qualitative description I just gave you. And so here, the volumes reiterate that the grass carp have larger telencephalons and mesencephalons, while the big-headed carp have larger cerebellums and vagal lobes. This suggests that grass carp likely use their telencephalon to process and behave in different ways than the big-headed carps. Based on previous literature suggesting that fishes have some spatial cognition and learned motor behaviors housed in the cerebellum, it's possible that big-headed carps use more learned motor behaviors or rely on species-wide behaviors for success in prey capture and predator avoidance. An important additional feature of this is to see that the standard error bars are relatively small, revealing a large degree of individual consistency between the sizes of the brain regions within a species. So what does all this mean, and what else do we need to know to move forward? First and foremost, the overall three-dimensional shape and volume of the large-scale brain regions only tell a portion of the story. To supplement this information, I've also looked at the cytoarchitecture across the brain and found some very interesting interior shape and neural density differences between the species. Unfortunately, I don't have time to show you all of those results today, but I'm happy to chat about it with you, chat about it during or after the conference. What I want to leave you with today is that we have found that there are interplays between phylogeny and ecology that shape the brain in these fishes, as well as how variation in the brain between the species may result in differing abilities to succeed in a changing or new environment. Having this atlas as a resource can tell us all about how these animals perceive and may interact with their environment. Hopefully for the next ABS, I'll have some more details of this story to share with you. Thanks.